So in the last lecture, we were using the sequences file, FastDrive file, and the file of the genome that we are using to align that reads. Now we want to extract the annotations of that file. We want to extract the features from that file. So we need a genome annotation file, which is also a GFF and general feature format file, or a GTF file, which is gene transfer format. So gene transfer format is a file format used to hold information about the gene structures, how genes are aligned there, how genes are represented there. It is a depth delimited text format based on general feature format file, but contains some additional conventions specific to the gene information as well. So it contains about 9 columns, which first column contains the position of the chromosome, second contains the sequence, and third contains its feature. It's if it's a gene, if it's a transcript, if it's an exon, if it's a CDS, it's a UTR, whatever it is. Column 4 and 5 contain the start site, end site, and the column 6 contain the header, and the 7 contain the strand which is represented on, 8 contain the position, and 9 contain the IDs that are represented there to the ensemble IDs, I think so. So that was the for the main GTF file. So after knowing about the GTF file, I think we should look at how to get the RNA expressions. So now that we know we need a GTF file, so we are going to download a GTF file, which is a Saccharomyces cerevisiae GTF file, which I'm also going to show you how you can download according to your own genome. So here is the genome reference annotation in GFF, GenBank, or tabular format. So if you need in the GFF, like I told you, GFF, GTF is the same format. Just click on the GFF and it should start downloading just like this. So it's a 54 megabytes of file for the human genome. After that, we need to index that BAM file. So we are going to index that file, which I already did. I think we have done it in our previous lecture. Source.bam file was created, I think so. Get source.bam. Yep, source.bam was created. We already have the BAM file. We already have done the indexing format. We already got the genome. So let me just show you the genome. I'm gonna get Saccharomyces. If I can just find the file here. Yep, get Saccharomyces cerevisiae genome. So that is the genome annotation file. So I'm gonna terminate this operation and make a head of first 100 lines of Saccharomyces cerevisiae file. So here you can see the positions and all the stuff that I told you behind, if it's a gene, transcript, whatever it is. So that is for you the GFF file. Moving on to the slides, how to get the extraction features out, how to extract that kind of features, how to extract our RNA expressions, how to check the expression pattern of our gene. So the primarily intended use case for the HTSeq-count is differential expression analysis. So what is the primary purpose of the tool that we are using? It's called HTSeq. It's a high throughput sequence analysis in Python, where one compares the expression of the same gene across the sample and not the expression of different genes within the sample. So now we consider two genes which share a stretch amount of sequence such that for the read mapping to this stretch, the aligner cannot decide which of the two genes of the read originated. So hence a report for the multiple alignment is created by HTSeq-count. So HTSeq-count is the command that we are going to use for our extraction, features extraction, we can say so. So for that, we are going to install our HTSeq. Just keep in mind we need Python for using the HTSeq. So sudo apt get install python.ht-seq is the command for installing our ht-seq. I'm just gonna see if I already got in my system because I'm not working on this right now. Yep, I don't think so that I got this python.ht. So let me just run a command. So sorting the same file using the, so we are also have to sort the same file for some reason. So I'm gonna sort this as well. So while it is sorting, yes, align.sam is already sorted. So I was just checking it. I need to I need a HTSeq-count to create a sort.m file. So what kind of files that we need? HTSeq-count is the command that we need. Format is the full. BAM is format that we are using. Sort.bam is the file which contain our expression. And the GTA file is saccharomyces file that we need. So here is the sort.ham file creation. So if it runs, yep, it did. So HTSeq-count was already installed in my system. So what it does, it analyze the 4,000 41,879 GFF lines processed. It indexed and couldn't retrieve the index files or .ham. Two SAM alignments were processed. So it is saying it didn't find any files so .ham. Yep. So there wasn't any sort.m files. It created a sort.m file which contained all the number of genes that were there in the reads. So these were the number of genes that was there. So I just gonna get sort.ham file to a our what we can say expression dot txt file. So get expression.txt file. Yes. So we got the alignment. There were no features because we are only using the two reads. There weren't any significant matches to that reads. So here is this expression. So there aren't any expression of our genes. There also all the values are 0. You should have known what fpcam value is for this lecture because in the previous session I told you about. So here the number of genes that are analyzed. So there were no feature that were found because we are only using the two reads. If we have used the file around 400 megabytes and something like that, there will be a lot of features that we can. So just for the sample, just for the demo, I used only two reads. So our command line section is done. We have 28 slides. It was done easily and quickly. So that was it for this lecture. If you have any questions about the command line section, about the graphical user interface and something like that, you can still ask me. So that was it for our lecture of gene expression analysis on command line and graphical user interface.